afternoon and welcome to Radcliffe. My name is Janet Rich Edwards. I am the faculty director for the Life Sciences programming here at Radcliffe and an epidemiologist at the school across the road there, across the river. I particularly want to welcome you to this, our second lecture in the Epidemics Science Lecture Series. If you haven't picked up one of these hot pink cards there in the way back when you're on your way out, in addition to this terrific lecture that we have today, we're also going to be talking about the epidemic of poverty a week from today. Later on, obesity, more complex than you think, and Alzheimer's. So these promise to be, I think, really exciting lectures, and we're certainly glad to have really very qualified and thrilling faculty, including Dr. Louise Oaklander, who will be talking today about small fibers and big pain. She's Associate Professor of Neurology at Harvard Medical School. She was trained at Rutgers and at Johns Hopkins, has both an MD and a PhD in neuroscience. She directs MGH's Nerve Unit and a federally funded research team at Mass General that studies peripheral nerve disorders. Her team has discovered a new neuropathy common in children and young adults and its first treatment, which is an immunotherapy, and she'll talk about that today. They've also linked fibromyalgia to small fiber neuropathy. She served on advisory panels for the NIH, the FDA, and the Institute of Medicine. I will take a moment to tell you how we're going to run. Dr. Oaklander will give her talk, and then we're going to open up to you guys for about a half hour of questions. We have a roving mic that can come to you, and I would just say at this point, while Dr. Oaklander, I'm sure, is a very good clinician, this is not going to be a clinical consultation. So, you might think now, if you have sort of questions about personal things you've experienced, how to phrase it in a way that is more general and applicable to everybody. So I encourage you to do that. And I think with no further ado, let's welcome Dr. Oaklander.
Thank you, Janet. Now, what was that word you used? Was it thrilling? Thrilling? Did she say thrilling? Wow, that's a tall order. And I guess it's appropriate because I understand that there are people here from even other states. Did anybody come here from another state, drive a long distance? Where are you from? New Hampshire. New Hampshire, okay. Not that far. How about far off states? Wisconsin. Okay, does she get the prize for farthest driving in cold weather to come hear this talk? Anyway, I'll certainly try to be thrilling. And it's always hard to talk about science with a mixed audience. Some of the people here are faculty members or graduate students. Some are members of the public. There's people who know a lot, people who don't necessarily know a lot, have a lot of interest. I'm going to try and see if I can straddle things a bit. But don't worry if you don't understand every single word on every slide. It's intended sometimes for trying to satisfy those who might have great curiosity. So I'll start at the simple beginning, which is what are nerves and what is neuropathy? That's what we're talking about today. The peripheral nerves is the part of the nervous system that's outside of the brain and the spinal cord. That's what a lot of people think about when they think of neurology. What we're talking about today is the wiring that connects our CPU to the outside world. When we take a cross-section through a peripheral nerve of a human, we can see these small fibers, which are the kind of nerve axons that I'll be talking about today. These are these tiny little thin guys. That's why they're called small fibers. There's a bunch of different ones, including C fibers, A deltas, and autonomics. Because of these many different functions, as you'll see, polyneuropathy can have different medical causes and cause different symptoms. The functions of these small fibers are so diverse that when people develop neuropathies that affect them, they can just develop a lot of different symptoms. And we're going to go over some of those symptoms, but I think there's an underlying problem, which is that a lot of patients who have this kind of neuropathy it's not even picked up. They're searching for answers. They know they've got problems in more than one organ system. People haven't put the pieces together. It's like the blind men and the elephant. So let's go over some of the symptoms. And again, not every patient has every symptom, and I'm not able to talk about all of the symptoms, so I'm just focusing on the most common ones. And clearly one of them is chronic widespread pain. That means it's not like you fell down and banged your knee and you have pain in your leg, but it's really pain affecting both sides of your body for no clear reason or no better explanation. And pain from small fiber neuropathy comes in two varieties. So the most common is so-called distal pain, which means it starts in the feet and the hands. And I've shown you a picture of a woman, a patient of mine, who has the erythromelalgia phenotype, which means that in addition to having painful legs, they're also red and they're swollen. So when we see those three things together, the Greek for that is erythromelalgia. It's just a particular flavor of small fiber neuropathy. Not everybody with erythromelalgia has this neuropathy, but at least 50% do. And for those of you with a particular interest in this, there is May 9th Erythromelalgia Awareness Day with events at the State House on the other side of the river, including yours truly. But not everybody has pain that starts in the feet. About 1/4 of patients, it starts elsewhere, and you can see this woman has the erythromelalgia as well. Equally, perhaps even more common, are cardiovascular symptoms. Except people don't say, gee, you know what? I woke up this morning and I got cardiovascular symptoms. What is it that they actually say? They say, I woke up this morning and I can't get out of bed. I feel lousy. I feel terrible. Maybe I'm coming down with something. But the problem is when it goes on day after day, week after week, month after month, year after year, it's not just that you have the flu. Something different is going on. And it can be very difficult to pinpoint what exactly the cause is. The kind of cardiovascular problems that are most common is, you can see here, this poor guard standing on duty for hour after hour has fainted. Something called orthostatic hypotension, which means people can feel dizzy or actually faint, typically when they're standing up, sometimes after meals. Another one is just chronic fatigue, inability to do your normal routine. We're going to talk more about that in the next slide, but I wanted to particularly draw your attention to my colleague David Systrom at Brigham, who specializes in this, and he knows more about this kind of causes of fatigue than anyone I know. So he's the one to talk to. Patient education, which I suspect there are a few of you in the audience who, maybe some of you know your patients, maybe some of you might find out you are patients from this talk, education is just critically important, because very often there's so much stuff you can do. If you just understand what's going on, you can control things yourself. And in that sense, patient advocates and patient support groups are very important. I see Leslie Levine here. Can you raise your hand, Leslie? Who runs one of the local support groups, so she's a resource. And I wanted to point out Dysautonomia International. Can you folks raise your hand as well? Are you national or international? International. Support group for patients. Do you have some literature in the back? That sounds great. And they're not even paying me anything to say this, truly. In fact, yeah, definitely not. And so these are some of the things that patients can learn to do to control their symptoms and improve them themselves. I think first and foremost is the use of support stockings. And I like that photo because it goes to show that support stockings don't all have to be ugly. So here's some science behind this for those of you that are so inclined. And this explains, I think, the best explanation for why people with neuropathy can have this kind of fatigue and inability to get out of bed and do their routine. And these are microscope slides, unfortunately not out of my lab, but out of a very skilled colleague of mine. And what these shows is muscle cells under the microscope. I think they're best seen here. But the focus is not on the muscle cells themselves, it's on the blood vessels that bring the blood, the oxygen, the glucose, the cookies you just ate downstairs on the way in, to the muscles so that the muscles can do their job. And the problem that these patients have, the muscles are still there, but what you can see in the slides from the top with a healthy person is there are all these axons, these nerve cells, that come running down and they control the blood vessels. See this round blood vessel here? The little yellow squiggles are the nerves that tell the blood vessel when to open and close. And what you can see is in the slides from the bottom, there's far fewer nerves and the blood vessel is gaping open. So the point is that when your nerves are messed up, they control the blood vessels, your muscles may not get the oxygen and nutrients they need to function. And in many people, not all, that's why they can't get out of bed in the morning when they have neuropathy. This is an emerging area that I think is of great importance, hasn't been addressed, and that's the fact that small fiber neuropathy can actually affect the brain as well. And so it happens in a variety of ways. They haven't been adequately studied. I hope there might be some brain scientists or experimental psychologists here in the audience who might become interested in taking this up themselves and looking into this. This picture that you can't see is just to show you formal cognitive testing that's done on patients who have this kind of problem when they're lying down flat and their blood can make it to their brain, and then they're on a tilt table. The tilt table stands them up vertically, just as I am now, and their blood can't make it all the way up to their brain. And so their brain function decrements, and that's been captured in testing and published. Gastrointestinal symptoms are very common as well. This is a microscope slide from my lab. Heather Downs, where are you, Heather? Raise your hand, Heather. So Heather Downs is the person who made this slide for you, this beautiful image that shows you these are the intestinal crypts, and all the little squiggly green lines are the small fibers. So you can see that your gut is under the control of these nerve cells. And I think that explains why many patients develop all sorts of symptoms, upper GI, lower GI, and they go to see a gastroenterologist, but in fact it could be neurologic. In the worst case scenario, some patients lose so much weight from intractable vomiting that they have to be nourished with a nasogastric tube, or some of them even by vein. Treating these symptoms, they can be treated. And again, if you know what you have, if you get the tests that are recommended, there's stuff you can do to make your gut feel better and calm down from neuropathy. This also is to show that it's not worth doing MRIs. A lot of patients with neuropathy have MRIs. The only MRI finding here is that this little boy's colon is full of feces, again, because he can't move his bowels because the nerves that control the bowels aren't working right. The bones and joints even are innervated by these nerves. Orthopedists, immunologists don't think of it that much, but the bone marrow even is densely innervated by this particular type of small fiber nerve endings. You can see here in one of my patients with a hereditary neuropathy that he's even had loss of the bones in the ends of his fingers. In order to track all these symptoms, there's so many that we realized we had to develop our questionnaire. There was no questionnaire anywhere in the world. And so with the help of my former postdoctoral fellow, Roy Treister, we developed this questionnaire. We're continuing to improve it, right, Gary? With Gary Zerpeli. But we've published at least one paper that shows reasonable validation and performance of this. And these are questions, you can't read it, but this is something that is increasingly available. This captures the symptoms we've heard from our patients, not just what's in the textbook. Okay, so what are the tests that can confirm small fiber? As you can see, the neuroexam is not that sensitive and may not pick it up. The usual tests, EMG nerve conduction study, don't detect it. Even nerve biopsies may not pick it up. So skin biopsy is really the test that we recommend. And again, Heather Downs is the person who manages our skin biopsy lab. These biopsies sound worse than they are. They're done under local anesthesia, but they need to be done by somebody who knows what they're doing. They can be requested to be performed at Mass General, or a doc locally can do them and ship them into Mass General for analysis. Either way, the instructions for this are on our website. Max Klein and others from our group have done a lot of work on the normative data. And these are studies that depend on normal volunteers. More than 400 of them have come to our lab and allowed us to remove a tiny bit of skin and measure the nerve endings. And I show this to you, showing you these more than 500. What you can see is these normal people vary a lot in the amount of nerve endings they start with. Normal kids, the red marks here, have far more nerve endings to begin with than normal adults do. And the problem is that a lot of the commercial labs just use one threshold, and if the count that's measured in you is above the threshold, they say, fine, you're normal. If it's below the threshold, they say, oh, you've got neuropathy. But it's really much more mathematically complex than that. We've done modeling, and so when you have a skin biopsy, please ask which lab your biopsy is being sent to for interpretation. And for young people, our lab is the only one that I know of anywhere in the world with accurate norms for kids and teens. We're big fans of physiology, which means how the nervous system is functioning, not necessarily what it looks like under the microscope. The tests there, autonomic function tests, as far as I know, Mass General and the Brigham are the places that offer them here in Boston. They measure a bunch of different aspects of nervous system function. Sweating is particularly important, something called the Q-SART, and this person here has loss of normal sweating from the ends of their legs. So let's turn a little bit now that you know the symptoms and you know the tests for neuropathy, let's turn a little bit to some of our discoveries. And neuropathy, anybody here who's a physician in the audience will agree that what we were trained is that neuropathy affects older people. Diabetes, cancer, chemotherapy, these are the common causes. So one of the things that's come out of our lab that I think has been very useful is the information that neuropathy affects kids much more often than anybody realized. And we knew that there were some rare genetic causes. Our lab is very interested in genetic causes of neuropathy, and insofar as I know, Mass General is really the place to come for families with these kind of neuropathies. We have a nerve gene family clinic where we can see entire families. You can see these 3 very nice boys that are photographed here. We're interested in other genetic neuropathies as well, and this shows another family we follow that has HSAN1. Remember I showed you the dad and his terrible hand up there? We're seeing his daughters in our clinic as well, with the idea of trying to treat their neuropathy before it gets so bad. Karen, are you here? Karen Weintraub? Karen's done it. Oh, there she is. She's lurking in the back. She's done a terrific job of helping us to bring this to awareness, and there's more information on the STAT website if you're interested. But how about non-genetic causes? Everything I've shown you before is very rare. Again, kids were not thought to have neuropathy because they don't have the medical causes of neuropathy. And I believed the party line until I met this college student from New Hampshire who got admitted to Brigham with severe pain out of nowhere. Very healthy guy, but look at those legs, right? That's that erythromyalgia again. And we figured out that in him it was small fiber neuropathy, but we really had no idea why he should have it. All of his tests came back normal. On a hunch that it might be autoimmune, we started treating him for that, and literally within an hour or two, his pain started to improve. So this was truly what's called the index case, and it changed my career, and now look at how many other people are hearing this story. So the importance of one patient's story. Well, one patient is not proof of anything, and so with Max Klein, there you are, my right-hand person, we went back and looked at the records of all the young people we'd seen with the onset of chronic pain before age 21. And Max and I published this paper in the journal Pediatrics in 2013 that really laid out this new disease that had never before been described. And again, you can see some of the characteristics. 3/4 of the patients were girls or women. Some of them were grown now, but they'd had the onset of pain before age 21. And again, look at all the different kinds of symptoms they have. You guys know all this stuff. Oh, I can skip this slide now. It just shows you that a lot of these young people had abnormalities on their skin biopsies or on their autonomic functioning tests that really showed objective evidence that they had neuropathy. So we looked at these young people to figure out, what was it, 41, I think? To figure out what the causes were in them. Well, none of them had the genetic diseases that you just heard about, and they didn't have family histories. What came out is that although some of them were said to have psychiatric problems, in fact, and had hospitalizations, none of them really had serious psych diseases. But what they did have is a history of autoimmune illnesses, or when your immune system begins to attack your own body. That paper was published in the pediatric literature, but it's important to realize that there are a lot of these people who began to be sick during their teen or early adult years, and they may now be in their 40s, 50s, or 60s, and they could still have this and still improve with treatment. So we next started with prospective studies where we specifically recruited people and matched controls to further test this hypothesis. And we chose to study fibromyalgia. Anybody have fibromyalgia here? Anybody been told? Wow, okay, so this is of interest. Fibromyalgia affects 1 to 5% of the population in the literature, and there's a consensus case definition, which makes it good for research. We looked at all the different measures of small fiber neuropathy we had at our disposal, but we particularly prioritized the objective test results because they can't be faked or exaggerated. And that study showed that 40% of the fibromyalgia patients versus only 1 in the control group had objective evidence of neuropathy. Kind of hard to get that one published. What I was saying sounded so strange because no one's really found any biological underpinnings until then. But of course, what's happened since is I know of more than a dozen studies that have looked at this, and they all are reporting the same thing. Doesn't cause all of fibromyalgia, but what we would say a substantial minority. And again, there is a review article that came out in Scientific American Mind that talks to some of the different investigators. This study is for the technicians in the audience, and it explains how small fiber neuropathy can cause pain, and that it's similar when they look and study patients with fibromyalgia and small fiber, they find the same electrophysiologic abnormalities, where these pain neurons, they're normally only supposed to fire if somebody's sticking a knife or a pin in you, but they're firing spontaneously. Those nerves are active. They're sending pain messages up to your brain, even when nothing painful is actually happening to you. And those pain neurons also fire excessively and in a prolonged way. This other very technical slide from my colleague Frank Rice from Albany shows evidence under the microscope that the arteriovenous shunts, again, the blood vessels, are abnormal in these patients with fibromyalgia. And again, it's the blood vessel abnormalities that we think underlies some of the symptoms we see, like erythromelalgia. With Max, we've turned our attention to another unexplained, similar, vague illness known as Gulf War illness. That's still a problem now for people who were in the Gulf War, in the first and second Gulf Wars in the early 90s. And we have several grants from the DoD, and Max's study shows strong evidence that some of the veterans, not all of them, with Gulf War illness probably have this same underlying early onset small fiber that came on at the time they were military recruits, and it's persisted. The next step, if you're diagnosed with neuropathy, is to figure out what the cause is. And again, indeed, there are a lot of different underlying causes. We recommend that patients speak with their doctor about getting these blood tests performed. These are the ones that are supported by published papers in the medical literature. We've looked into this ourselves, as I'll show you. And it's really important to figure out what the cause is in each person so you can treat it. This blood test checklist is available on our website. When we When these tests were administered to the young people, we didn't find evidence that they had diabetes or heavy metal toxicity. The only thing really that came up were blood tests that are associated with autoimmune conditions. And the majority of the patients, the young onset patients, did have immune markers. When we step back to look at the value of these blood tests in patients of all ages who have idiopathic neuropathy. What does idiopathic mean? Sounds like that's the cause of it. That's the kind of neuropathy you have. If a doctor tells you that you have idiopathic anything, just say no. That's not a diagnosis. Idiopathic is Latin for we are idiots. All it means is the doctor doesn't actually know what the cause is in you. And so they turn it into Latin, and then the patient thinks, oh, I've got a diagnosis now. I've got idiopathic neuropathy. Not so much. Press your doctor for the real diagnosis. We looked at these blood tests in patients with idiopathic small fiber neuropathy, and what we found is that we could pick up an underlying medical abnormality that could be causal in more than half of them. Very surprisingly, it turned out that diabetes and high blood sugar, which is said to be the most common cause of neuropathy in the US, was not common in this population. The reason for that is that people who have diabetes and who develop burning and tingling in their feet or nausea, they kind of know that they have diabetic neuropathy, and they're not labeled as idiopathic. In fact, again, we found evidence of unsuspected immune blood test abnormalities. Well, all this medical stuff and research, it's all fine and dandy, but at the end of the day, what the patients want, what the doctors want, what even the insurance companies want, is treatment. That's really what it's all about. And so the information that we've gathered from our patients has enabled us to start treating them. In the study of the young people, it wasn't everybody who got immunotherapy, only 15 among that very small group. The immunotherapy helped about 80% of them, so very high level. Let's talk for a little bit more about autoimmune neuropathy and what this is, what this concept is that we are really bringing to public attention. So the job of the immune system is to protect our body from outside invaders, like the flu. I think every one of us knows that the flu is going around. But what happens sometimes is those immune cells can be fooled and mistakenly attack some of our body's own cells, mistakenly thinking that they're invaders. And there are autoimmune diseases that affect pretty much every cell in our body, everything from rheumatoid arthritis to multiple sclerosis to vitiligo, you name it. There's autoimmune diseases. Because the nerves and the brain and the spinal cord are so important, they're actually hidden behind a barrier, an immunologic barrier, which protects them. But these small fibers are in a very interesting situation. Since their job overall is to help defend us against threats, they actually go outside the walls and they stick their cell bodies and they stick their distal axons outside of the barrier, and they are so in tune with immune cells, they actually secrete immune mediators, and they have physical contact with some of the immune cells, in particular mast cells. This shows you the dorsal root ganglion, which is where the cell bodies, the CPU, if you will, of these small fibers is located. And as you can see, it's outside of the spinal cord, so not protected from autoimmune attack. What I'm telling you, I stand up here, I'm a Harvard faculty member, it makes perfect sense, but the stuff I'm telling you is not in the medical textbooks. And so, it's controversial, but what is it I'm supposed to be again? Exciting? Thrilling. Okay, so I got to tell you guys stuff that's not in the medical textbooks, and you judge for yourself whether what I'm saying makes sense to you. What is in the medical textbook now is really only 2 immune causes of small fiber neuropathy, of which a rheumatologic disease called Sjogren's is by far the most common. The major symptoms of Sjogren's are dry eyes or dry mouth. So if you have dry eyes or dry mouth, along with neuropathy symptoms, it's something for you to consider. But even a lot of doctors don't realize how common polyneuropathy can be the presenting symptom of Sjogren's in up to a quarter of patients. And a lot of the patients with Sjogren's are what's called seronegative, meaning their blood tests may not even show it. There are also very rare immune neuropathies that are associated with cancer. But hey, in our most recent paper, which studied 55 patients, a quarter of them had autoimmune, known autoimmune diseases, but 3/4 of them didn't. So what the heck is going on here? Well, I think it's thrilling. I think it's thrilling because I think we might be discovering a new disease. Maybe you guys can help. We really haven't found a good name for this. So when we write our NIH grants and our papers, we have to be very conservative, and rightly so. And so what we're calling this now is apparently autoimmune small fiber neuropathy. And see, I can't even say immune because we're just starting to do the basic science studies that are needed to prove this. So I'm calling it apparently autoimmune. But this takes up a lot of space when we write grants. So if anybody has a better name, if you could come up to me afterwards and tell me your suggestions. And we're involved now in a whole series of papers that we're publishing with collaborators inside and outside of the lab. How about that celiac paper, Maddie? You're going to do it, right? Okay, I've got a commitment. Showing the overlap and links between various types of autoimmune disease and neuropathy. There are different kinds of autoimmune diseases, and the evidence so far suggests that autoantibodies may be, and B cells, which are made by B cells, is probably more important here for a bunch of reasons. You don't see a lot of cells when you do spinal taps or when you do biopsies from these patients. And there also are detailed blood test abnormalities, for instance, in the complement pathway, that are most consistent with B cell immune-mediated dysfunction. Although clearly when you have long-standing cases, helper T cells, without any question, are involved in sustaining that. So putting this all together, again, treatment is our focus. Our most recent paper reported on the first ever study of immunotherapy for apparently autoimmune small fiber neuropathy. This is a retrospective study, meaning it's a look back at patients who were already treated. It's not the so-called gold standard, the placebo-controlled randomized trial. The reason for that is because those cost millions and millions of dollars, and you got to get the money from NIH or other similar funding agency. And the problem is they are not going to give you the taxpayers' money to do this kind of study unless there is some published evidence in the literature that you're barking up the right tree and not the wrong tree. So we've now published the paper that I think shows some evidence that it might be reasonable to consider these kind of trials. This is what's called a proof of concept research publication. This was done with my former postdoctoral fellow, Dr. Liu, and we looked back again at patients who had received a particular treatment called IVIG, which is the best treatment for other types of autoimmune neuropathies. So we had given it to some of our patients here. We selected them very rigorously. We do not want to give these treatments to people who have other causes of neuropathy. That wouldn't work, and it would be a lot of time and effort and wasted money. These are very expensive treatments, I should mention. We tracked how patients did. The symptom important to a lot of patients was pain. We tracked objective measurements from AFT, autonomic function testing. The difference in AFT results is shown here. The blue are the abnormal AFT results before going on this treatment. The red is after. So 93% had abnormal results before starting the treatment. 54% had abnormal results after treatment. And the adverse events were pretty similar to use in other illnesses. So we're building on something that we call the small fiber registry. And what a registry is it's when you go to a doc and they really try to track the information they get from you, and they try to collect the information in a standard way. And it's a heck of a lot of effort to develop this. Again, it's not something that is grant funded, but you kind of need it in order to do research. And yeah, I'm looking at the epidemiologists in the audience who would agree. Any other epidemiologists here, by the way? Yeah, okay, so I got to just look at you two. And this just shows you some of the types of information that we have. We're very interested in, very bullish on the samples, the blood samples. And if any of my patients are here who've donated our blood, we're incredibly grateful to you, because this is a tremendous resource for future research. This goes to show you some of the kinds of experiments that we do with this blood. I think the most important experiments overall are the ones that involve something called passive transfer. And this is what's used to really prove whether a disease is autoimmune or not. And it involves taking a frozen sample of that patient's blood. We're able to take extracts of that patient's blood and inject it into little lab animals, and then see whether those mice or rats develop symptoms of neuropathy. And if we do, that's something that we can capture in our lab as well. And that provides proof that in that patient, they have an autoimmune cause or something in their blood that's causing their neuropathy. This kind of research is being done more and more in the cancer field, where patients' own samples are used to create individual mice or rats that then can be used to test the effects of specific treatments, like specific chemotherapy, what's going to work in that patient. So this is a very powerful technique. And what you can see here is the rat that was injected with blood from the patient shown on the bottom. It's the serum from the blood, not all the blood. Developed some behavioral abnormalities, some signs of pain in the paw of that particular rat, whereas another rat shown in blue maintain normal sensory function. So again, we're going to see differences in the different animals depending on which patient's serum was used. We're also developing standardized exam forms, and that's a project in particular of Sean Downs. There you go. With the guidance of Gary Zerpeli, and this allows us to figure out which aspects of that become abnormal are most useful for diagnosing neuropathy. And all these tests I'm telling you about, all these fancy tests, is wonderful for the people who can come and get them, but we are well aware that 99.9% of the people in the world can't come here to Cambridge and Boston or similar universities to have these kind of tests. So what we're trying to do is we ask the people who are coming for these fancy schmancy tests to also see if they're willing to let us take a few non-invasive, simple tests and see how the results of that correlate. The idea is that if we can develop simple, easy, cheap tests and show that they work almost as well as the expensive tests, then they might be able to be used all around the world. And with Max, who's an engineer, and some of our other collaborators, Dr. Serador from Rutgers, we're looking at the pupil responses in the eye. The eye is a window to the nervous system. Some people say the eye is a window to the soul, but I prefer to think of it as to the nervous system, because those same nerves control the functions of your eye. And so you can see this eye from one of my young patients is very dilated. Her pupil is very dilated. With these pupillometric goggles, what we're able to do in our lab is to measure these pupillary reflexes, record them, to test whether these are useful things. If it turns out to be useful, it's something that could be very widely used. For instance, perhaps maybe it's something that could be put on a cell phone. There aren't any biotech entrepreneurs here, are there? Just checking. That's proprietary. So I'm going to wrap up by stepping back and bringing this up to think, well, I've told you about rare genetic diseases. I've told you about common diseases, diabetes, a lot of different causes of neuropathy. How big is this problem anyway? Well, here's a clue. This is the Epidemics Lecture Series. So I've run some sort of hand-waving thought experiments. No one knows the numbers. No one knows the right answer here. But if you look back at what's been published, there's really only one epidemiologic study so far out of the Netherlands. And if you apply the numbers they got to the world population, it suggests there might be 4 million cases. of small fiber in the world. Only problem with that is for that study, you had to have a physician-verified objective diagnosis. And what I've been telling you is that more than 90% of the people don't even know they have this disease, and they've never even seen a neurologist. So it may be that there's closer to 40 million cases in the world than 4 million. But here's another way you could look at it. Fibromyalgia affects several percent of the world's population. Given that a dozen labs are pretty much showing that maybe 40% of these may have small fiber neuropathy, well, then that yields the idea that there may be 75 million fibromyalgia patients around the world who could have small fiber. But not all small fiber patients have been diagnosed with fibromyalgia. So when you go back and you factor the rest of them in, well, you know, now you're starting to look at numbers in the hundreds of millions. And again, I'm not saying that that's how many people have this. We really don't know. The information just isn't there. But whether it's 40 million, 4 million, 400 million, either way, this told us we need to start looking at this at the population level, not only just with the individual patients that make it into my office. And so for that reason, we're collaborating with epidemiologists at the Harvard School of Public Health. And Gary and I, with our collaborators, are getting ready to publish the first ever epidemiologic study. Now, this is on all peripheral neuropathy, not just small fiber neuropathy, in the US. And this is through the Nurses' Health Study 2, which has been tracking hundreds, over 100,000 nurses all across the US for decades. And we have impeccable information about their healthcare records, which one has diabetes, which one was treated for cancer, what drugs were used in many of them. And we've been able to pick out more than 3,000 people who say, yeah, my doc has told me that I've got peripheral neuropathy. So by going and sending out special additional surveys to them, we anticipate that we're going to be able to learn more about peripheral neuropathy than has ever been known anywhere in the world. That's the power of epidemiology. Okay, so to allow time for questions, I'm going to give you a small summary first, and then we're going to move into the question period. So I'm going to give you a small summary of a big topic, that the symptoms of small fiber neuropathy include pain and itch, fatigue, POTS, things that people don't associate with neuropathy. I've told you what some of the recommended diagnostic tests are, that blood tests are very useful in identifying, not in everybody, but in the majority of people, in providing hints as to what the cause might be. Sometimes the tests need to be repeated more than once, or some people may need specialized genetic tests or other special tests to figure out what's going on. That there's strong evidence from labs all around the world that patients who have, whether it's CME, CFS, Gulf War illness, chronic Lyme, that a substantial proportion, somewhere between a third to a half of people who receive these labels may in fact have an actual underlying potentially treatable medical disease. So that's maybe the single most important message, is to take this information out. Every one of us, I think, has family members or friends who are suffering from these illnesses. It's really important that they know that they can ask their doctor, have we thought about this? Would it be reasonable to think about testing me for this? Some of the patients with this type of neuropathy probably have immune causes, and in them, the treatments for other immune neuropathies are quite reasonably safe and appear to be effective. We don't know the numbers of people who have this around the world, but either way, it's a heck of a lot. Either way, it's too many. And my point is let's step away from this whole opioid crisis business and giving people painkillers. I got nothing against painkillers, but we're so much smarter than that now. And we can find better treatments that actually fight back at the disease, not just masking the pain that you have to take for decade after decade. So let's move on to the era of smart medicine treatment. And I'd like to extend my thanks to Radcliffe for this wonderful opportunity, this auditorium, the publicity. By the way, the cookies downstairs are fabulous, so thank you for the cookies. I'd especially like to thank my patients because, again, what I'm telling you is not stuff that's in textbooks. We're writing the textbooks, and we're writing the textbooks because of what we've learned from our patients. So all of those people who've let me ask them questions, who've spent more than an hour in my office talking to me, thank you so much. Thank you to the members of our research team, past and present. I'm presenting the work of dozens and dozens of collaborators. And thank you also to our funders, both the National Institutes of Health, the Department of Defense. We have several people and private foundations who've made substantial donations to our group, which is very important, something we can put directly to good use. And I'll end by mentioning that a lot of the information I've told you is available on our website, our Neuropathy Commons website. My phone is telling me something, probably that it's time to end. Thank you again for your attention. And
Perfect timing. All right, so we have some roving mics, so go ahead and, oh, lots of questions already. Great.
I'll leave it up to you.
No, you go ahead.
So why don't we start in the back of the room for a change? Who's farthest back? Is that you?
Thank you. So in the beginning of the talk, you mentioned that GI symptoms were frequently a symptom of peripheral neuropathy. And then at the end of the talk, when you were talking about autoimmune, you mentioned something like celiac-related neuropathy. So I'm wondering, how do we know that the GI symptoms are a result of the neuropathy as opposed to the neuropathy being a result of a GI problem?
Great question. I mentioned celiac because it's autoimmune, but only a minority, only a few patients actually have celiac. We test for it routinely because so many of these people have GI symptoms and because people can have celiac and have no GI symptoms. But it's only a very rare cause. Maddie here is going to write the paper with me that'll define how common it is. It's pretty rare as a cause. From the back with the pink shirt.
Is there any relationship between the mechanism of the post-herpetic herpes pain and the small fiber?
Absolutely. That's not a plant, by the way. Absolutely. And so I actually started my research career at Johns Hopkins, where I trained, and my first research grant was on post-herpetic neuralgia. And our lab showed that post-herpetic neuralgia is associated with severe damage to these same small fiber neurons, because again, they're the neurons that transmit pain signals. And shingles or zoster specifically damages sensory neurons. And so yes, we view post-herpetic neuralgia and also post-herpetic itch, which my group put on the map, as kind of a restricted form of these same, I'm pointing here because this is the most common location where people get shingles, as a restricted form of a small fiber neuropathy. And by the way, in case you haven't heard, there's a very effective new shingles vaccine that's going to be coming to the market within the next few months. And it's dramatically effective even in older people. Not that that's any of us here. But if you know anybody who hasn't been vaccinated against shingles, now's the time. I'll let you, I'm not very good at picking, so I'll let you do the picking, and I think there's time for everybody to get their questions answered.
Hi, thanks for your talk. I was wondering if you had any thoughts on chronic regional pain syndrome being a diagnosis related to small fiber neuropathy. And one more, any thoughts on amino acid peptides? And my friend next to me has questions too.
What was your second question?
My second question was about a treatment being peptides. Amino acid peptides? No?
So I don't know. That's simple. No, I don't know about amino acid peptides as a treatment.
And the first one was
The
CRPS
first one was about
or
CRPS.
RSD. Yeah.
And after we wrote papers on shingles, then we turned our attention to complex regional pain syndrome, which has a lot of similar symptoms, the unexplained pain, swelling, color changes, changes in hair. And we published the first objective evidence that that is what we call a regional small fiber neuropathy, where only part of the body's nerves are affected. I've been telling you about polyneuropathy, which means it's a generalized nerve problem. So yes, we think CRPS is very closely linked, and that it's a regional small fiber neuropathy. And furthermore, CRPS, which is when people have a kind of an injury, you know, a skiing accident or cut their leg or a sprain, it's something that would just get better, and for whatever reason, it just doesn't get better, and the pain and problems can go on for years sometimes. We think that in some of those patients, they may actually have an underlying small fiber polyneuropathy, and it's for that reason that their nerves aren't able to recover after they've been injured.
Thank you.
Yes, I had a question. It was a really great lecture and it's very informative. So in terms of the skin biopsy that you do in patients, and have you had any sort of insight in terms of if it's done once on a patient in the beginning of their symptoms and their symptoms are progressing and the initial biopsy is not showing anything, what is your protocol and what happens to patients who are progressively getting worse? Would you repeat it soon, or how do you observe those patients? And the second question was about when you mentioned...
the first question is already... let me just answer the first question first. So the question is about skin biopsies, and that's currently the best test available, but it's far from perfect. There's no medical test that's 100% perfect. And so what I would say, if the skin biopsy shows that you have severe nerve damage, then it's highly likely that you do have this kind of neuropathy. That's called positive predictive value. But on the other hand, it doesn't always pick it up. As you say, what if the biopsy is done early in the course? Or what if it's taken from the wrong part of the body? So the fact that you have a normal skin biopsy doesn't mean you don't have this kind of neuropathy. It makes it less likely. And some patients I've been following for years, their skin biopsies were normal at the beginning. We would suggest they might want to repeat it a few years later if we still didn't know what was going on. And in some of them, it's dropped over the years. It might still be in the normal range, but it might have started out at the 70 or 80th percentile, and it's dropped 60, 40, 20. So that tells us their nerves are going or not healthy, or it drops into the abnormal range. So they're not perfect, but it's still very important. It's a very important, very useful test.
Okay.
And what was your second question?
The second question is about Sjogren's. You mentioned that a lot of patients are seronegative. How do you sort of correlate patients who might have the autonomic testing showing positive and the sweat testing, but then they're seronegative? What is the next steps? And they're very symptomatic.
That's kind of a complicated question because the sweat testing isn't the recommended test for diagnosing Sjogren's. So Sjogren's requires a rheumatology evaluation by somebody who's really familiar with it. They factor in a lot of things, the symptoms you have, looking in the eyes, there are formal diagnostic criteria. Sometimes they take a biopsy from the lip to look at the salivary glands, and it can take a few years to work out a Sjogren's diagnosis. Should we work our way towards some of the patient people? We can come back to the back, but I want to also get some equal representation from the front.
Hi, I'm a patient, and I just want to understand better the impact on the brain.
Could you raise your hand again? Oh, great, thank you, okay.
I'd like to understand better the impact on the brain sort of both ways. One of your slides mentioned polypharmacy as a potential cause. So I don't know if that's too many medications impacting the brain, and then also, could occipital neuralgia be a headache caused by small fiber neuropathy?
That's a whole complex topic. I don't know of anyone. I'm a neuroscientist and I'm in a neurology department. I'm surrounded by experts on cognitive function, by experts on headache, and I don't know of any of them that's looking at small fiber neuropathy yet. So another one of my goals, I hope I have some colleagues and fellow researchers in the audience. Another reason to give this talk is to excite people who are working in different areas to start taking up their own projects. we'd be thrilled to have experts start to look at this. What I tried to summarize in that complicated slide is that peripheral neuropathy, specifically small fiber, can affect the brain in a lot of different ways. For one thing, those cells, we think of them as peripheral, but they actually send part of their cell up into the spinal cord. So it's part of the central nervous system as well as the peripheral nervous system. And they have even the ones that don't send an axon up the spinal cord, but that end where they come into the spinal cord, they synapse, they connect with other nerve cells. So there are transsynaptic effects. And then there are network effects in the brain when the brain cells change their function, for instance, because of reduced sensory input coming in. And then what you mentioned is what I call the downward spiral, which was when people are sick and they don't know what's going on and they're doing all this stuff and they get on a medication for this, for their stomach, for their pain. They become depressed, they become inactive. All of these things have effects on the brain as well. And so those really are tertiary and quaternary effects of being so sick. So it affects the brain at a lot of different levels.
Thank you.
Hi, I was asking about ESR because you use that as one of your tests, and I've heard that it's kind of unreliable, and I was wondering what sort of ranges you're looking at and why you chose that test.
We actually don't rely on any one test of autoimmunity. It's not a black or a white situation, for exactly the reason that you point out, which is that these tests that are used to track immune function, I wouldn't call them unreliable, but I'd call them nonspecific. And so that means they don't correlate that well. Somebody could have an autoimmune disease and have normal blood tests. A certain number of people have abnormal blood test results and they don't have autoimmune diseases. So they're statistically associated, but it's not a black or white relationship. And that's why I mention these things like, I don't remember the exact percent, have abnormal ESR, which stands for erythrocyte sedimentation rate. Others may have low complement. Others may have a test called ANA, anti-nuclear antibodies. And again, none of these are specific for small fiber or any particular disease at all. But together, they kind of give a hint, a clue, when they're elevated in such a high percent of patients that there may be a link to autoimmunity, not in each person.
Hi, thank you for the talk. I wanted to ask, you mentioned CFS or chronic fatigue, and it's one of these idiopathic things that might have small fiber neuropathy as the underlying cause. What sort of work has your group or others done on that?
My collaboration with David Systrom and his team at the Brigham, I think, is going to publish some really exciting data coming out of that. And I think it's okay for me to mention it since we've already published it in preliminary abstract form. And what that's going to show is that a substantial percent of people who present, he's not a neurologist, he's not a nerve doc, he's a lung and circulation doc. People who go to him because of this lack of energy and just difficulty getting out of bed and inability to do their routine, substantial percent of them do in fact have this kind of peripheral neuropathy. So not everybody, but enough that a doctor should always think about it as part of their differential diagnosis. And it's treatable. Once you know, I mean, there are many reasons not to be able to get out of bed. Depression, for instance, is a very common one. So it's important to go beyond the, I have chronic fatigue, to let me go see a doctor like Systrom who can do the test to actually measure my blood flow and my heart rate and my blood pressure under different circumstances and hone in on what's the exact cause in me, because he's got effective treatments.
So I wanted to ask sort of more broadly, we have about half of patients with POTS and chronic fatigue syndrome and fibromyalgia, the syndromic disorders, have a tangible small fiber neuropathy. And you're looking at the immunotherapy in these patients. Has anybody...
In some of them.
In some of these patients, right. Has anybody looked for like functional receptor antibodies that maybe don't cause a neuropathy or haven't yet at whatever stage in the illness, doing like the passive transfer work to, you know, rabbits or something with serum from patients who don't have the neuropathy to see if the immunological profile you're finding might be in those patients too?
Yeah, we would always use control sera, and so it's very important to thank not only the patients who come for research, but also the normal controls and people with other diseases, because all of the work we do, we have to compare what we find from patients to what we find in the general population. And so, yes, we would always use serum from non-patients to do these kind of experiments as well.
I had 2 observations I'd like to share. It seems that the cause of this problem is in the plasma, and that it's an immune complex problem. There are immune antibodies that are in the plasma. Has plasmapheresis been considered as a treatment, rather to remove the offending plasma and replace it with colloid or clean plasma, instead of infusing immunoglobulin G to downregulate the production of autoantibodies? Perhaps plasmapheresis could remove the offending agent. That's one observation. The other observation I had was that, or question, there's talk about genetic involvement. Has consideration been given to the abnormal gene sequences being viral inserts and not inherited from a prior infection, DNA from a particular virus, enterovirus perhaps? So if it lies in the genes in that sense, that would account for the chronicity of the illness, and that would account for the remissions and then the relapses, because the genes are still there expressing themselves. So I'm wondering, I guess the point is that has consideration been given to the fact that these might be gene inserts, not mutations, and not inherited? And then
Great question. Great question. So actually a couple of questions and comments. And one is about other types of immunotherapy. And again, immunotherapy is only one treatment, and it's a treatment for those people who, it turns out, we think have an autoimmune cause, which is really not everybody. But for immunotherapy, you have to think and individualize the treatment for the different patient. And there are different immunotherapies. There's no one size fits all. And so yes, absolutely. I do have some patients who use plasma exchange, taking out their own blood. It's like dialysis. It dialyzes away their autoantibodies. But again, another expensive therapy that's very hard to get approval for. It's got its own problems. And so it's not something that is routine. We try to start with the simpler, more mainstream immunotherapies. There's a whole host of new immunotherapies that are coming to market. Eculizumab, for instance, is newly approved for a neurologic autoimmune indication. So it's really exciting days. And that's why it's so important that basic scientists get involved in this so that we can actually figure out what the molecular epitopes are, what the targets are of antibodies, because when we really know what's going on at the molecular level, then we can use much more targeted therapies. As you say, IVIG is kind of a dirty drug that is a very crude way of approaching it. It's the best treatment we have now. But I think as this comes into sharper focus and as other research teams get involved beyond my own, it's going to open the door to much better therapies. With regard to the genes are involved. Without any question, genes are involved in autoimmune diseases. We're interested in looking at the HLA antigens. Some people have just unknown genetic mutations. I was at NIH last week working on a family with a novel, very clear familial genetic cause that's never been described in the literature. So by doing detailed mapping of the genes from that family, the affected individuals, the unaffected individuals, we're likely going to discover a new gene that causes neuropathy. But even in people who don't have familial neuropathies, we know that tendencies are genetically inherited. And so when we can get our database up to the level of tens of thousands of samples, right, Gary? At that point, then you can really start doing, population genetics, epigenetics, and much more sophisticated genetics.
Hi, thank you so much for your talk. I have two questions for you.
Thrilling?
I said thank you so much for your talk. Yes, it was thrilling.
Thank you.
And my sister just texted me very excitedly about your slides being posted on the website. I'm sure she will find it as thrilling, if not more so. So she's actually the one who has sent me along with two questions. The first one is, you mentioned IVIG as the primary therapy, but are you familiar with subcutaneous immunoglobulin and if any of your patients have used it as an effective treatment?
So the question is, what's the relative roles of the same treatment, which is pooled human IG immunoglobulins taken from blood donors, such as us, healthy blood donors, comparing it when it's given intravenously, which is what IVIG stands for, as opposed to subcutaneously, which is SCIG. There are pros and cons of giving it either way. They both get it into your body. They're both effective. It's really a logistic issue, and there are different logistics. Different insurance companies approve it in different ways. By the way, I'm telling you this, that we published a paper that it's reasonably effective, as if that means you can then go out and get it. But you need to, maybe you know already, but for the rest of the audience, is that the insurance companies pretty much don't approve these treatments. And it's very rare that they will approve it. The reason is they don't come to lectures like this. There aren't any insurance, medical insurance. See? I told you so. I told you so. And because of that, they may not know the things that are emerging from the leading research universities. And so therefore, even though things are published, when your doc tries to get approval for treatment, whether it's subcutaneous or IV, pretty much, typically, the response is no way. That's why we put our paper on the website. And if your sister's interested, she needs to get a copy of that paper, because you have to physically take the published evidence, give it to your own treating doctor, and have your treating doctor send it to the insurance company and say, well, hey, now there is evidence that it works, so please consider it.
Thank you. And the second question was, could you tell us a little bit about your thoughts on the relation of mast cells or mast cell activation disorder to neuropathy?
I'm very interested in that, but it's such a complicated question that let's defer that to the discussion time afterwards. Yes, very important, very interesting.
Hi, thank you. I have one question that may just be restating what you've said, so I apologize if that is all it is, but just to clarify, if you had a patient who, say, had a diagnosis of POTS and fibromyalgia, just as an example, and maybe they were poorly controlled, is that enough to say, let's test for this? Or are there specific physical symptoms that they should have as well?
That's a great question. That is a great question. And I think again, it's an emerging area. It's very unclear. We don't know for sure who will not have this. We don't even have, as with the skin biopsy question, there's no perfect test. The good news is that there's an upcoming meeting that'll bring together experts around the world to develop what's called a consensus case definition. And when there's a case definition, it's very, very helpful, even though they're not perfect either. But at least what it means is there's a mutually agreed upon diagnostic standard as to who has it and who doesn't have it. So that's going to be very helpful. Well, you know, fibromyalgia. If I were a fibromyalgia patient, I mean, I think patients want to get better. And when you have an illness that's really a label, that's the key point, is that fibromyalgia is not a diagnosis, it's a label. It's a word that we put on a collection of symptoms. It doesn't have one cause. Those same symptoms have different causes in different people. And it's so important to move beyond the label and press your doctor to tell you what's the underlying medical cause in you. And even if they might not know it in 2017, maybe they could figure it out in 2018 or 2019. So I think it's up to each person with fibromyalgia. People have, unfortunately, different levels of medical coverage. Some docs are more interested in pursuing things than others. But, you know, I know what I would do if I were the patient.
Thank you. I just had one other quick observation.
Sure, let me just, okay? we've got time for one or two more questions. And in the interest of side of the room parity and gender parity, I also want to invite this gentleman here who's had his hand up since the beginning. Kristen, maybe you can put your hand up again. Why don't you ask your last question? Well,
okay. And I'm happy to stick around. So if you didn't get your question answered in public.
This, I hope, is a fairly short question. I'm interested in the dynamics of the pathology. In other words, if you have a patient with a positive skin test who responds...
Could you speak a little louder? Hold the...
If you have a patient with a positive skin test who responds to immunotherapy of some sort, how rapidly does the skin biopsy return?
Great question. We don't recommend repeating skin biopsies more often than once a year because the pathology, as important, as vital as it is, changes slowly. Then it takes a while for the nerve cells to regrow. And they may not even regrow in everybody. And they may regrow at different rates in somebody who's 15 than somebody who's 65. So as of now, the skin biopsies are most helpful for initial diagnosis and their role in tracking patients. In fact, we're just applying for an NIH grant to try and better clarify which are the outcomes that are most useful for longitudinal tracking, as opposed to initial cross-sectional diagnosis. Good question. Worth waiting for.
As was this lecture. So I think I have to cut off the questions officially for the group and let people get out who need to get out. But I know that Dr. Oaklander is going to stay up here and can answer a few more questions from you personally. And I just want to thank you again. This is just terrific stuff.